Hello, Mr. Allman here, and I want to share with you a little bit more today about the plasma membrane. Now, it had been long known, at least long inferred, that there had to be some type of barrier between the inside of the cell and the outside. Otherwise, there wouldn't be any reason for the stuff inside the cell to stay inside the cell. The problem is that the microscopes for a pretty long time were too stinking poor to be able to actually see anything because the membrane is super small. It's something on the range of a few nanometers thick, and a nanometer is times 10 to the ninth, times 10 to the negative ninth meters, and so we're looking at something like a thousandth of a micrometer. And so because that they were so thin, there was no way to be able to see these things for several hundred years after the time that cells were first really observed and described by those early compound microscopes. But with the advent of electron microscopes, a lot more within the cell was really able to be deciphered. And one of those particular structures that we now are able to see as chemically one of the most interesting parts of the cell is that of the membrane and the other parts of the membrane that are chemically related to it. So we'll come back and look a little bit at the function of the endomembrane system, but first we need to understand its structure, and for that we're going to switch over to the macromolecule prezi to get a little bit more detail on what it is that this is really made of. So macromolecules are those things that we are found, types of molecules found within living systems, and lipids is one category, and a lipid is a biochemical term for a fat. Now we know fats are things that will repel water. So oils, waxes, those are all things that will not, if you put them into water, they will separate away from it. They will form droplets in the water because chemically water is polar because it has those partial positive and negative charges, but lipids don't. They're nonpolar. They're completely neutral throughout their the entire length of the molecule, and so that's a really important point in understanding how it is that cell membranes do their job of separating the inside of the cell from the outside world. So to skip ahead now to phospholipids in particular. Phospholipids are a type of modified triglyceride. A triglyceride is it consists of a glycerol, which is a short carbon and hydrogen-oxygen molecule, along with 3 fatty acids, and those 3 fatty acids are long chains of carbon and hydrogen. Now, in a phospholipid, we replace one fatty acid with a phosphate group. So you can see the phosphate is this part added at the top. The phosphate has a phosphate ion, along with choline, which is a small chain of carbon, hydrogen, and nitrogen. And because of the phosphate ion, phosphate has these negative charges on it, and so phosphate is hydrophilic. It will interact with water. The fatty acids, of which there are 2 in a single molecule, the fatty acids are long chains of carbon and hydrogen, which are hydrophobic. So this is an amphipathic molecule. It has a hydrophobic tail and a hydrophilic head. So it is really half and half. So in the same way that soap is amphipathic, because soap will attach to the grease and dirt that's on your hands because of its hydrophobic part, but soap will still be washed away when you run soap under water because part of the soap is hydrophilic. So in the same way, the cell is mostly hydrophilic. So the interior of the cell is a water-based environment. The cytoplasm is a water-based environment. And the exterior of the cell most often is a water-based environment as well. Cells, whether it's a multicellular organism surrounded by fluid that is water, or single cells that are floating around in the environment, come in contact with a lot of water. And so we don't want the water-based stuff inside getting out. So a layer of fat provides an effective barrier because water cannot pass through. Water is repelled by the fat, and so it's that chemical repulsion keeps the stuff inside allows it to stay inside. Now, there's a lot more to the cell membrane than just the phospholipid. Some other important parts. There is cholesterol. So cholesterol is another type of fat. It's a chain of 4 rings of carbon, hydrogen, and oxygen. And the job of cholesterol is to stabilize the plasma membrane. If it gets really cold, the cholesterol is going to help the plasma membrane to stay fluid, so it doesn't freeze. If it's very hot, the cell membrane makes sure that the 2 layers are going to stay with each other and they're not going to separate from each other. Other things that are in the membrane include proteins. Proteins of which there's a few types. There are channel proteins that have space in the center for materials to pass in or out of the cell. There are integral membrane proteins, those that kind of span across the entire protein. There are surface proteins and peripheral proteins that are attached just to the exterior or just only partially wedged into the membrane. And each of those has a particular function, again, transporting materials in and out, serving as chemical signals, or in the case of the peripheral protein, acting as a place where the cytoskeleton can be anchored to the to the membrane. The other significant features in the membrane include glycoproteins and glycolipids. You see the glycolipid has the set of orange circles on it. The glycoprotein has the blue circles on it. Those circles all represent carbohydrates, and carbohydrates can all be arranged in different patterns. So you see that there are different shapes in which those carbohydrates are arranged. Those shapes act as signals, and so the type of shape that is found on those glycoproteins and glycolipids sends a signal to other cells or sends signals to the immune system, letting other parts of the system know what type of cell this is. So they're very important in terms of chemical messengers. Now, the arrangement of the phospholipids is in a double layer. So we call it the phospholipid bilayer. So there is a layer of phospholipids on top and a layer of phospholipids on the bottom. And you notice the arrangement is always consistent here of the hydrophobic tails pointing in towards the center and the hydrophilic heads pointing towards the outside. So what it's creating here is a sandwiched layer of fat. The fatty acids are staying in the middle because they are repelled from water, which is on either side of it, but the hydrophilic phosphate head will interact with water, and so it's positioned itself on the outside. So that's the general structure. Now let's switch back and take a look at the function here of this particular system. So there's a relationship here between all of these parts, and we're starting towards the nucleus and working our way out, because the idea that we're looking at is how it is that the cell can get stuff outside of the cell, if the cell membrane really is acting as a barrier that's supposed to keep those things inside. So one of the things that the cell wants to get rid of are proteins, and this is not a waste, but this is actually an intentional type of sending a material out of the cell. The proteins that are attached to the ER are known as bound ribosomes, and so ribosomes are making proteins. The proteins that are made on the RER are designated to be exported from the cell. You can see in the electron micrograph, you can see the structure of the ER is the same as the structure of the plasma membrane. Those 2 dark lines represent the hydrophobic heads on either side of the layer of fat, which is in the center. So again, it's a phospholipid bilayer. Each of the circles represents ribosomes. So you see there are free ribosomes and then bound ribosomes, which are on the RER. Now, the endoplasmic reticulum is the site of where many of those ribosomes are located. It's a network of membrane channels. It's the same type of membrane, a phospholipid bilayer. And you see it's folded, and the RER is always very close to where the nucleus is, because the nucleus contains the DNA. DNA has the instructions for how to make proteins, so it's convenient for the cell to have those things located right next to each other. You can see the RER here on the right, which has the ribosomes on it, That's why it's called rough, because when you look at it through the microscope, you see all those bumps. Whereas the smooth ER doesn't have the ribosomes, and so all you see is the curves. You don't see the ribosome, the bumpy ribosomes. And the purpose here of the rough ER is to create compartments within the cell that have particular materials within them. So, for example, proteins that are made can be surrounded within a compartment of the ER, and then that compartment can actually separate away from the ER into a transport vesicle. Now, these vesicles are simply, as you see in the green circles here, these are little spheres of membrane. There's membrane all around the outside, and on the inside there is any number of things. Waste products from cell respiration, proteins that were made in the ER, fats, and everything else in between. And so these things are being stored for later use in the cell, or they're being transported away. But chemically, they are really one and the same as the ER. It's a piece of the ER that has been pinched off and is now free floating in the cell. And when they're transported, they're going to be pulled. They're going to be pulled along the cytoskeleton from one place to the next by something called the motor protein. Now, one of the places where those vesicles might transport to is the Golgi apparatus. Now, the Golgi looks very similar to the ER because it is essentially the same thing. Structurally, it's the same, it just has a different job. So a folded, flattened set of phospholipid bilayer that receives vesicles. Those vesicles have materials inside. The vesicle will merge with the Golgi because the membrane surrounding the vesicle is the same as the membrane in the Golgi. And so those things will just come together. The stuff inside will enter into the Golgi, the center of the Golgi. And then one of the things that's often done, things could be modified and then packaged to be released again. And so usually they're given a new signal. Again, the signal is dependent on what the shape of the glycoproteins and glycolipids on the outside of the membrane. They are given a signal and sent somewhere else. You can see in the microscope image here, the receiving side on top, the membranes, flattened set of membranes in the middle, and then all these vesicles that are ready to go somewhere else in the cell, those are on the bottom. So the way you can tell the difference between the smooth ER and the Golgi is the presence of all of these vesicles on either side of the structure of folded membrane. Now the plasma membrane, again, phospholipid bilayer, and it is the same as those other parts. So when something like a vesicle comes in contact with the plasma membrane, it can actually join together because the stuff that makes up the membrane is the same as the stuff making up the vesicle. The function here, it's controlling what comes into and out of the cell. And it's also responsible for sending stuff, like signals from one cell to the next. So if we just review that again, if we have a material, a waste material, or a helpful protein that needs to be sent out of the cell, the ER will produce or package up those materials into using a little piece of membrane to surround it that will pinch off from the rest of the ER, forming a vesicle. The vesicle will be transported to the Golgi. The Golgi will receive those materials, package them up again, and provide a signal for the cell for where they should go, either to be stored in the cell for later or to exit the cell entirely. So we can imagine these little dots, if they represent waste that was produced by the mitochondria, that waste is being, in this case, it is being packaged, sent, merged with the cell membrane, the vesicles merging with the cell membrane, so that the waste is then pushed entirely out of the cell. And that process is called exocytosis, and we'll cover that a little bit more with active transport in a future video. So again, if you have any questions, please let me know, email, or talk to me in class. Okay.